All right. Welcome back, everybody, for the afternoon session of the first day of May 2021. My name is Guillaume Lajoie. I'll be the chair for this afternoon's session. Just a couple quick announcements here. So this afternoon, we have the pleasure of having amazing talks. The first is going to be by Dr. Bing Brunton, second from Dr. Alex Williams, afterwards followed by Dr. Maria Kimur, and followed by a keynote by Surya Ganguly. And after that, we'll have a student panel discussion on the career path in neuro AI. So without further ado, let me introduce the first speaker of this afternoon. Dr. Bing Brunton is joining us from the University of Washington in Seattle. Actually, quick announcement, I believe there is an in-person watch party organized at UW in Seattle. So hi to people at the Center for Computational Neuroscience over there. So Dr. Bing Brunton is an associate professor in the Department of Biology at UW. She also holds an appointment at the Paul G. Allen School of Computer Science and Engineering and at the Department of Applied Mathematics. Her training, which spans biology, biophysics, molecular biology, neuroscience, and applied mathematics, includes a bachelor's degree in biology from Caltech, a PhD in neuroscience from Princeton. Her group develops data-driven analytics methods that are applied to and are inspired by neuroscience questions. She has received an Alfred P. Sloan Research Fellowship in Neuroscience, the UW Innovation Award, and the AFOSR Young Investigator Program Award from 2018 for her work on sparse sensing with wing mechanosensory neurons. And today she'll be presenting us and talking about tracking turbulent plumes with deep reinforcement learning. Bing, floor is yours.
Thank you so much, Guillaume. And hi to everyone who is watching, wherever you are. I'm delighted to be here and for the opportunity to share some of our really recent work with you. So I'm going to start by sharing my screen. Guillaume, can you confirm that this is looking okay to you? No, okay.
Not yet.
All
oh,
right.
Coming. We're good. Yep.
Something is happening.
Now we're good. Perfect.
Now you see my title slide, hopefully?
Yep. All is good.
Okay. Fantastic. Awesome. Let's get going. All right, so I'm delighted to be here and to share a few minutes with you all. And so I'm going to be talking in particular about a very scoped talk today about one particular project. But I want to take a couple of minutes just in the beginning to talk more generally about the type of work that my group is doing and some ideas that I've been thinking about at this intersection of AI and neuroscience that I find to be exciting. We'll come back to it at the end of the talk. So in general, my group is I'm a computational neuroscientist. I self-identify as a computational neuroscientist. And so we have the pleasure of working on a variety of different model systems with a lot of diverse experimental collaborators. And so what pulls it all together is this ethos of developing data-intensive methods to understand neurocomputations, especially underlying naturalistic behavior. And so the students and postdocs in my lab work on a lot of different organisms, including insects and different types of birds, mammals. I also have a set of people who are concentrating on natural behavior in humans. This is a picture of my daughter Abigail in her natural habitat, doing what she loves to do best, which is eating spaghetti. I don't actually study her, but I needed a picture of a human, so there she goes. And to do all of this, I was thinking about these types of interdisciplinary meetings, which the main meeting is, and thinking about why it is that we build models like we do in computational neuroscience. And so for me, computational neuroscience and modeling in neuroscience in general is this really great connector in between disciplines. And not only does it connect disciplines, I think in particular for me, it connects a lot of really interesting ideas, like if you have multiple experimental results, especially across different scales. If you have genome level experiments and behavioral level experiments, I think computational models is one of the ways that you can really rigorously formalize the connections between these types of different experimental results. And the models are also really good. In fact, think oftentimes necessary for connecting the observations from diverse organisms. If you're interested in the same type of behaviors and class of behaviors, and you have observations from related or not so related organisms, I think computational modeling is a really powerful way of connecting these observations. We can also connect hypotheses with design of future experiments. So this is possible to do without actually formal mathematical models, but oftentimes our hypotheses, especially with neural circuits, with recurrence, gets so complicated that you really need a rigorous framework for connecting the hypotheses, building testable predictions, and then using those to think about what future experiments we'd really love to do next. And then finally, but certainly not least, computational models in neuroscience are really crucial for connecting neurobiology with mathematical theory and engineering. So I'm reaching in both directions here, both towards theory and mathematics, as well as engineering in terms of robotics and things that actually can be built in engineered systems in the real world. And so in thinking of making all of these connections and building computational models, it's a couple of things that I think about a lot when I'm building computational models. And these are things that I believe to be important in computational models in neuroscience. So the first is that I think about the brain as a way of the organism interacting in the real world with other things in the world, including the physical world, as well as other organisms, either of the same species or not in the real world. But it's for controlling and coordinating this behavior. And as such, I like working on natural behaviors. And I'm increasingly in my lab thinking about comparative approaches of thinking about neurocomputation. The second thing I think about a lot is that the body matters, right? Because if the brain is for behavior, then it matters what it is that you're controlling. It matters the physical shape and morphology of your body. All of your sensors and actuators are in fact embedded in this body. And so the biomechanics and biophysics of the body actually does matter. As such, we think about morphology and mechanics in a real way. And we think about embodied computations. And that's a point that I like to come back to at the end of this talk. And the third point I like to highlight is that feedback is quite central. This is, I believe, to be a great central feature of all biological systems, not just neuroscience and behavior, but the fact that there is so much feedback at every scale, from the level of proteins to the level of feedback with the physical world, feedback in neural circuits, and the feedback with future generations on evolutionary timescales. And so this feedback makes it so that a lot of what my lab has increasingly been working on are integrated sensory motor models, not just working on sensors or sensory or motor systems, but integrating these models into the same one. And really thinking about reinforcement learning as a paradigm for understanding natural behaviors, which is what I'm going to be highlighting here today. And so this feedback loop is really at multiple scales, but it's fundamentally an interaction between the animal through actions that affects the real world, which then changes in some way. Again, this includes social interactions and other things that can happen involving other organisms in the world. And that comes back to the animal through measurables. And the particular behavior that we'll be focusing on today is plume tracking behavior, especially in turbulent plumes. So I'm going to break down what all of those words mean in particular. So let's say that you have a compost heap. There's a delicious, very rotten strawberry in your compost heap, and you're a fly, so you think it's delicious, and you really want to go find it because you want to eat, and you want this to be a place where you might find future mates and provide for food for your future progeny. And so this behavior has been incredibly well studied, and the path through which, especially flying insects, will localize and then approach and then find these sources of odor can be described very briefly by the cast and surge strategy, which is, again, very well studied. I'll talk a little bit more about it later. And the strategy is briefly that in the absence of odor plumes that you can detect, you cast about, right? You're looking around for something to smell. And once you detect some kind of odor, you then surge upwind. And that strategy, as simple as it seems to be adopted by a variety of different flying insects and is very successful in localizing turbulent plumes. And so that's an observation from biology. And there's a lot of remaining questions about it. And so I'm going to be spending a bit of time digging into the particular results of this preprint, which is highlighted here. There's an archive preprint if you want to read more about the details. The work was led by a very talented a former graduate student, because he's now defended his PhD, Satpreet Singh, and is in collaboration with our collaborators Flourish van Bruegel at the University of Nevada in Reno and Rajesh Rao, who is at UW in computer science. So, like I said, this cast and surge strategy is something that's been pretty well described, both experimentally and in some theory. And it is valid in this regime where there's an odor plume in the air and you're some kind of small flying agent, flying insect. But this casting about strategy is actually more general than for odor plumes in the air. We also see it in tracking of terrestrial odor trails. I'm showing you examples here from 2 experiments of a rodent tracking an odor trail on the ground and an ant who is also tracking an odor plume, an odor trail on the ground. But compared to tracking terrestrial odor trails, tracking a plume in the air actually has some unique challenges as a task. So in particular, tracking odors if you are in a turbulent plume is difficult because the odors are intermittent. There's really not a trail, and there's not really a gradient or derivatives that you can really compute because the odors are kind of coming in these unpredictable little packets. The challenge of this task becomes even more challenging when the wind directions may change unpredictably. So you can't actually count on the wind coming in the same constant direction. And the plume statistics can also vary in other different ways. And so to solve this task, what the agent, the fly, and the animal actually has to do is a multi-sensory integration problem. You need to be able to detect odors, detect something about wind direction, and because the tracking of the odors is, the odor packets coming in intermittently, it also requires some form of memory. These are things that are necessary for talking the task. So like I said, I'm doing a very brief overview of the complexity of this task. There is actually a recent, actually upcoming review by the authors that are on the bottom of the screen there. It's coming out next year in 2022, summarizing the state of the art in this field of tracking turbulent plumes. And so experimentally, lots of experiments have been done on this behavior. So I'm going to highlight 2 sets of experiments that are particularly highlighting some of the challenges of this task. And so because this is usually some kind of a small animal that is going long distances, you really have to be able to track the behavior over these long time scales. It's not something that's really easy to do in a fixed tethered animal or in virtual reality. And so the sets of experiments that have been done include wind tunnel experiments. So you notice here on the left-hand side, there's a schematic of the wind tunnel experiment that is on the order of a meter long, which is the size of the arena you really need to get the convincing plume going and to then have the animal follow this plume. And if you really wanted to go crazy, you can do a long distance dispersal experiment. So this is an experiment that was done where the flies were released in the middle of the Mojave Desert and were then captured at a ring of sites that were kilometers away from the dispersal site. So there's a lot of long range behaviors, and like I highlighted earlier, the role of models. One of the complexities of understanding this is to reconcile the behavior with the neural recordings. And so if you have a really tiny fruit fly and it's flying over meters, if not kilometers, it's not really experimentally tractable to do neural recordings with modern technology that we can do. And so we can do some experiments and get a lot of exquisite experimental neurobiological detail on the tethered fly, or we can do these experiments either in a free-flying wind tunnel or out in the wild. It's not really possible to do both at the same time. And so the approach that we took in the paper that I'm going to be talking about is a complementary in silico approach. We are going to set forth a set of artificial agents and ask them to learn this task of localizing the odor source in the turbulent plume. And in this way, we hope to recapitulate some of the behavioral experimental results, as well as some of the neurobiological results of the underlying neural circuitry and the neural representations. Because in this artificial agent, we ought to be able to have access to both of them in a way that can hopefully synthesize and connect the behavioral experiments as well as the neurobiological experiments. And so going back to this picture of the animal interacting with the world and receiving feedback from the world, we're going to model the animal as an artificial agent trained with deep reinforcement learning. But in particular, I'm going to talk a little bit about our turbulent plume simulation, because that's the model of the environment is quite important here, as you can imagine, because we're interacting with this environment. So the artificial environment we have here is a relatively straightforward and very, again, computationally tractable turbulent plume simulation. The turbulent plume, there's an odor packet, comes from the cross. There's a little cross on the right-hand side. That's the origin of the plume. That's where the odor packets are coming from. The odor packets are spawned. They're born as a Poisson process, and then they follow the direction of the wind. Again, in some simulations, the wind direction can actually change mid-simulation. So they're following the plume and they're following the wind. As they go downwind, they're also diffusing. And becoming less concentrated in a Gaussian way. There's a Gaussian packet that then diffuses, and they're also jiggling around a little bit. So there's a bit of stochasticity in terms of they're not just deterministically following the wind, they're also jiggling a little bit in the orthogonal direction. And so that's our odor plume simulation. And what our agent has access to are, wherever it is in this arena, the local direction of the wind, so that's two numbers, that's a vector of the local wind direction, as well as the odor concentration. Okay, so there's also a reward signal, which I'll talk about a little bit later, that's needed for the reinforcement learning training apparatus. In return, the agent has the ability to move in the arena. So it has a speed with which it can fly forward, and it can also turn. So it has an angle that it can turn. And we implement this in the following architecture. The sensory inputs are made available to a recurrent neural network. And this is a vanilla recurrent neural network. The representation that is learned from this RNN is then fed into two different arms. So we have an actor-critic deep reinforcement learning architecture. Each of the actor and critic arms are multi-layer perceptrons. The actor then outputs the actions, the move and the turn. And then the critic is trying to estimate the value of this entire thing. And that is used in the learning process. So I'm going to show you what we get at the end, and then we're going to dissect some of these different components of it. What we get is the emergent behavior of the agent. On the left-hand side, we're seeing the behavior of the agent in this plume simulation. So you'll see here the plume is represented as a gray as the gray plume, and the agent has their trajectory outlined in blue and in green. It's green whenever it actually detects some odor, and blue when it does not. And on the right-hand side, we're looking at a dimensionally reduced representation of the neural dynamics of the recurrent neural network at the same time. And the two animations are synchronized. So you can kind of see that we now have access to the entire behavior repertoire as well as the underlying neural computations. Okay, so I'm gonna dig into this a little bit. So the first thing we noticed, to our relief and delight, was that the emergent behaviors of the agent looks very much like this cast and search strategy that has been developed in biological agents. In particular, we see that in the absence of the odor detection, the agent casts about. It kind of goes, you see it going orthogonal to direction of the wind, trying to look for the odor. And when it finds the odor, it goes upwards and follows the direction of the plume until it finds the odor source, exactly like you expect it to. So it has two behavioral modules corresponding to that. We call it tracking and recovery. Tracking for when it has the plume and is tracking the odor plume, and recovery is when it doesn't have the odor and it's casting about for it. There is a third behavioral module, which is the lost module. You'll see this on the bottom animation here, where it's basically making these tight corkscrews because it's utterly lost, has no idea what it's doing, and is just going around in circles. Sure, this is an RNN agent of a plume tracking behavior, but sometimes I look at that spiral and think it might be a metaphor for lots of our lives. This behavior that we observed is, like I said, recapitulates the biological observations of the cast and surge. And I'm going to make a small diversion here to compare it to a well-established and very well-studied algorithm for solving this turbulent plume tracking task called the infotaxis algorithm, which was popularized by Massimo Vargasola a few years ago. So there's a couple of similarities and differences between what we're doing and the infotaxis algorithm. So the similarities are that they're fundamentally solving the same type of task, and they are both solutions to a partially observable Markov decision process, the POMDP problem. And you'll see that at least on an abstract level, the infotaxis algorithm also has this casting and surging behavior, just like what we see in our agents. There's a couple of key differences, though. The infotaxis algorithm requires that the agent remembers a representation of the entire arena in terms of, and makes decisions based on a specific form of a Bayesian update, where it tries to figure out how to minimize the entropy of where the odor plume is. And so there are aspects of the infotaxis algorithm, at least the original version, and there have been a lot of innovation since, that it's unclear whether or not how this could be implementable by an agent that only has access to local information. Whereas in our deep reinforcement learning agent, like the one shown above, it only ever had access to the local information. And we can then dig into the neural representations of the RNN and figure out what, if anything, it's learning in solving this task. And so there's another cool emergent behavior that we observe in our agents that I'll highlight here, which is that it actually generates an interesting testable hypothesis that we hope may help resolve an ongoing discussion in the literature. And this has to do with what happens when the agents find the odor plume and what it's actually tracking as it's surging forward. And so the agent is a small object in this arena, and it is subject to the advection by the wind. It's following the wind. So if the wind is blowing in a certain direction, the fly will also go in that direction. And because of that, the fly's heading is actually different. Where it's headed and where it's trying to fly is different than where it actually ends up going. So its course direction, highlighted in this purple arrow here, is different than its heading, which is the red arrow. And so we can use this fact to try to ask this question: where does the fly actually go when it finds the plume? Because there's this ongoing discussion in the literature about whether or not flies are actually going upwind. What we can do here is compute the course direction relative to two different frames. The first frame is the current wind direction. So if it's following the wind direction, then we would expect that the course to be at plus or minus 180 degrees to the current wind direction if it's following the wind. And you can see in the plot in the top here that it's actually not terribly well aligned with the current wind direction. Instead, it's much better aligned with the center line of the plume. You basically draw a hypothetical center line through the entire history of the plume. It seems that the fly is actually following the center of the plume rather than the current wind direction. If the wind is not changing directions, these two are exactly the same thing. You can only really see it when the wind is changing directions, which is really why it's so important that we had this aspect of the simulation. In the agent that you're seeing on the bottom animation there, right there, you're seeing that because the direction of the wind changed midstream, there is a section of the tracking right there where you can see the agent going up plume rather than upwind. Okay, and on average, we can see this behavior. And so we're actually seeing that we're going to pose this as a testable hypothesis of the project, that the agents track the plume central line rather than the wind direction. We can also dig a lot deeper into the neural dynamics because we have access to this. And so we're giving the agents three pieces of information, which is the local wind direction, X and Y as a vector, and the current concentration of the odor. Everything else has to be represented and learned by the RNN. And so if you look at the neurodynamics of the RNN after the training, what we see is that there's some representation of head direction. And head direction is something that has been discovered it's actually a really wonderful field of discoveries in the Drosophila central nervous system, where there's been lots of discoveries of the representations of head direction in this ring attractor that is present in the central nervous system. We also find that there is some representation of the weighted average of the odor concentration, as well as a couple of other things that we think to be interesting because it requires memory, like steps since the last plume encounter. How many steps, how many DTs has it been since the last time the agent saw the plume? As well as the exponentially weighted average of the last time since the last odor encounter. And this fits a set of experimental observations that was recently published, where the orientation of a fly, this is now actually in the walking configuration instead of in the flight configuration, is more correlated with the encounter duration rather than the encounter frequency. So this is another thing that emerged from our project that seems to match really well with some experimental observations. So I'm going to drill down a little bit more on this idea of memory, because that seems to be so crucial here. Like I said, the intermittency of the sensory information that's available makes it so that it is not possible to compute gradients like in the way that T or chemotaxis would do. There is no gradient to be computed. And the odor encounters can be so intermittent that you may be going around for a long time without ever seeing one. And so intuitively, it makes sense that memory is a crucial aspect of what might be learned by this artificial agent. And so to drill that home, we did a set of experiments where we compared the RNN network, which is shown on the last set of dots right here, and compared it to non-recurrent networks, feed-forward networks, that are fed an artificial history of what was happening. So it either got access to the last couple of time steps, more time steps, or an even longer time history of the time steps. And what we can see is that among these feed-forward networks, the longer the memory, the better the agent does. And then the recurrent network consistently does even better than that. Under constant wind conditions, when the odor plumes are really sparse, and especially when the wind direction switches many times. So this recurrence, the success of the recurrent neural networks, as well as the feed-forward networks with even longer time histories, both suggest that the role of memory is really crucial in this task, which I think really jives with your intuition of how you might solve this task. We can also do some digging, more digging into the neurodynamics. Here we're looking at eigenvalues of the recurrence matrix in the RNN before training and after training. Before training, these are seeded as Gaussian random variables. After training, we looked among a couple of different agents, and they both, all of them, show this very interesting behavior. So we're looking at these eigenvalues on the complex plane, where the red dotted circle is the unit circle. And so we see that after training, you have at least one eigenvalue that is outside the unit circle that's strictly real valued. And there's almost always a couple of eigenvalue complex conjugate pairs that are outside the unit circle. And so exactly what this means is something that is ongoing work. We also looked at the eigenvalues of the recurrence Jacobian to look at the effective stimulus integration timescales that is being performed by the RNN. And we see here that before training, these eigenvalues of the recurrence Jacobian have time steps that are really vast. And then after training, they've mostly come down to a manageable time scale. And this time scale we believe to be indicative of the time scale that is necessary for solving this task. And so going back to this picture that I showed you in the beginning of this interaction between the animal and the world through actions and measurables. So this is something that we've been thinking a lot about. And beyond this one particular task of everything in the simulation I've shown you is pretty much scaled to the Drosophila in terms of ground speeds and plume statistics, stuff like that. But it kind of connects to a more fundamental question that we can ask, which is, why do we even have a nervous system at all? Because after all, even if you're E. coli, you can do this behavior of localizing odor sources, because localizing sources of chemical sensing and localizing sources of chemicals is pretty crucial, regardless of where you are on the size scale, right? So this is crucial for finding food, for finding potential mates, and for evading potential predators, for example. And that's something that spans a lot of different size scales of different organisms. And not all of these organisms actually have a nervous system, right? So the sensory and motor capabilities of the E. coli and how it does chemotaxis are also extraordinarily well studied. And this feedback in the circuitry there is implemented at the level of protein circuitry and enzymatic reactions rather than as a nervous system. And so it begs the question, why do we even have a nervous system if E. coli and paramecia can actually do this without a nervous system? And so what I would speculate is that this is something that has to do with what I would like to call, and many other people have called, embodied intelligence. The idea that the nervous system evolved to deal with the growing size scale of the organism and its interactions with the physical environment that it finds itself in. Right? So an E. coli and the whale not only are vastly different in size scales, but they also operate in fluid regimes with vastly different Reynolds numbers. And dealing with the delays that come with having to coordinate a body that is vastly larger in size with a lifespan of an animal that is decades different, that I think is a key clue to why we have a nervous system and why it is that we really have to be thinking about not only artificial intelligence, but embodied intelligence. And I think it's really going to be difficult to understand biological intelligence and artificial intelligence without really taking the body and the plant, in control theoretic speak, in a really serious way. So with that, I will conclude my talk and thank Sat above all, because this is all highlighting his last project as a PhD student and the rest of the lab. So thank you.
Thank you so much, Bing. This was a wonderful talk. So we have about 8 minutes for some questions, and I will read some here from the Q&A section. So one of the topics which I actually started out here, actually there's two with the same upvotes, but it's related to the use of RNNs and how related is it to the circuit structure in the fly, right? So in other words, there's some recurrence, and you've seen that that's key in sort of short-term memory. And there's other questions piggybacking this comment, saying that there's some known problems in training these RNNs. So how closely matching are the dynamics that are known from fly circuitry and what you obtain in your RL agents?
I think at this point in our first attempt, it's not at all, right?
Okay.
And nor was that a constraint, because the circuitry of the fly does a whole lot more than just find rotten fruit in the air, right?
Absolutely. Yeah.
And we've also made absolutely no attempt to constrain any of the circuitry based on the wealth of knowledge that we have now about the fly connectome. And I think that's actually one of the really exciting frontiers in this work is the emerging connectomes, not only at the fly brain, but also its ventral nerve cord and its spinal circuitry, right? The part that actually interacts with the muscles and the actuators of the wings and the legs. And so I think of this as an iterative refinement process, right? Yeah. Like these models we don't take them that seriously. We take them at the level in which they are designed. They make some testable hypotheses. We go back to some experiments and we can refine them. And so the key for me is that they acknowledge feedback in a serious way, not in a biophysically realistic way, not in a mechanistic circuit level way, but at least there is feedback. Right? We acknowledge the fact that there's feedback, like we know there to be in the real organism, and that feedback happens not only on the level of neural circuitry, but also with the external environment. The rest can be refined,
Fair enough. So actually, to follow up on this point, right? Like, not to have the biological realism, but there's something to be said in the sort of dimensionality reduction structures that, or the structures that can be revealed with dimensionality reduction. So there's a question by Paul Sazak here. Do you know if anyone, example, V. Jayaraman, applied dimensionality reduction to the Drosophila central complex and seen evidence of some of the kinds of relationships you found in your RNN, for example, odor concentration and etc. Right?
Oh, I don't know. I mean, there's been a lot of work on the central complex, and so but the fact that I don't know about it does not mean at all that it doesn't exist. It very well might. I haven't heard of it yet. But I think because we're talking about a behavior that clearly requires multisensory integration, it's not just the navigation module, right? There's also the sensory integration module that's pretty crucial here. And so, for example, wind sensing is something that's really crucial for performing this task. You can't do it without being able to sense local wind direction. So the wind sensing circuitry has been worked on by many people, including most recently, I've seen a couple of papers from Kathy Nagel's group, where again, this is the tethered prep, but that's all that you can do if you wanted to really dig into the circuitry of this, thinking about the wind sensing circuitry of the fly. And then how does that get integrated with the odor sensing, which again, lots of people work on the odor sensory capabilities of the fly, right? And that all at some point goes into some central circuitry, and then that then outputs to the wings. I mean, there's a lot of different pieces of this. And I think that's actually something I'm really excited about, is that there's a lot of detailed knowledge that can be synthesized in a model, but I don't think we know enough to attempt a full detailed model right now. Rather, I think we need to build a framework and then have it be sufficiently modular that we can put in pieces of known physiology.
Very cool. So maybe tying up the other end of the spectrum here, there's another kind of question topic, which is, about the metrics that you use to quantify similarity between the actual trajectories, like the end behavior between RL agents and insects, right? So in the question and answer, Sat actually commented on that question, saying how hard this is, right? And there's no kind of magical recipe, but I wonder if you could, comment a little bit on these observables that you try to match.
In the behavior specifically?
Yeah, like the trajectories, like you said you had some, following and correcting or something like that, right? Like, so are these things that are well quantified or just eyeballed?
Yeah, I mean, so what is in the project so far are, I would say, well-informed heuristics is what we've come to. We did not do this in let's say, an unsupervised pattern discovery way. We could have, and it's unclear whether or not that would teach us more, be more insightful than what we've done so far, right? I'm obviously a big fan of that whole line of work where you take trajectories and kinematics of natural behaviors and discover modules and motifs. There's a lot of exciting work on that. I think because we don't have a detailed model of the kinematics, and we've completely ignored biomechanics so far in this project, right? So that doesn't seem like it was it would have been a fruitful way to go. So we would have had to do that on the trajectories, and so that's something that we could have done. But I think I'm simple-minded sometimes. I think the modules we came up with they're the most visually salient ones to people who are watching it. And hopefully the couple of videos I've shown you have given you a taste of what that is. I think if the simulations became more complicated and rich and had more subtleties to it, that would certainly be an interesting direction to go. It didn't seem for us like it was necessary for what we've done so far.
Fair enough. Okay, well, I'll thank you again, Bing. I think in the interest of time, we'll pivot to the next speaker. But this was a wonderful talk and great lessons for embodied AI. And so we'll invite you to stick around and enjoy the rest of the afternoon with us. All right.
Thanks, Guillaume. Thanks, everyone.